From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 16th, 2024. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 63rd episode of Matters Microbial. I genuinely appreciate every listener and viewer of this podcast. Thank you all very much. Speaking of which, I received 2 emails at the Matters Microbial address I wanted to share. The first was from a professor at another institution thanking me for a conversation with a scientist, and that inspired him to invite that scientist for a seminar. That was very nice to hear. The second was from a listener or viewer that was hoping for another guest or two to discuss fermented foods like kimchi. And I agree it would be a lot of fun. I will be looking for such an expert. My Bio 350 Micronauts this week continued their work. As you can see, I did my best to light the way with this self-portrait using photobacterium. The Micronauts were able to begin examining the identity of their water bottle buddies, isolated from their water bottles previously. We had carried out colony PCR using universal 16S ribosomal RNA primers. And as you can see from this photo, most students obtained great amplicons. I'm very hopeful. We then cleaned up the reactions and sent them off for DNA sequencing. It's going to be such fun to determine who is who. The marine microbiological safari has continued as those same Micronauts restreaked and observed isolates from their seawater samples. Some isolates were typical, as seen here. Another was highly fluorescent under UV light, like this one. Possible siderophores? Of course, we're looking at agar-degrading pitter colonies seen here. And finally, microbial oddballs like this pigmented isolate that appears to be secreting a dark pigment into the medium around the colonies. We cannot wait to learn the identities of these candidates. Moving on to today's topic, we return in part to bacteriophages, viruses that attack bacteria. And here is a wonderful plastic model a friend sent me from Japan showing a phage attached to its bacterial prey. The history of oxygen in our biosphere is interesting to me. After all, oxygen is highly reactive, almost a poison. Yet we require it to live. Actually, it's an electron acceptor. Most students learn that plants produce oxygen, splitting it from water, and extracting that high-energy electron to harvest to power a cell. But in fact, the plants have chloroplasts that carry out this and other duties, which in turn are descended from Cyanobacteria. Cyanobacteria is an interesting name, and it kind of harks back to some confusion about the nature of these organisms. They were originally called, and I cringe as I say it, blue-green algae. They are not blue-green, nor are they algae. They are, however, bacteria. In any event, eons ago, a bacterium was first able to use sunlight in a fashion that produced oxygen as a waste product. These cyanobacteria changed the face of our entire planet and made our very lives possible. So I work hard in my classes to remind students that the energy-producing organelles in eukaryotic cells, chloroplasts and mitochondria, are descended from bacteria. These bacteria became obligate symbionts with a respectful nod to Lynn Margulis's endosymbiotic theory and make our lives again possible. So, I am very pleased to introduce Dr. Nathan Ahlgren to discuss the work that he and his co-workers do in the study of cyanobacteria, their bacteriophages, and related topics. Welcome to the Quality Quorum for today, Nathan.
Hi, Mark. It's great to be here. Thanks for having me. I'm excited to chat today.
I think it's a wonderful opportunity because I've always loved symbioses, and so any opportunity to kind of fold that in is pretty important to me. But I think that for the listeners and viewers, a good place to start is how you ended up where you are. Maybe spend a minute or so with that?
Yeah, sure. I think growing up, I was always interested in biology. I have an interesting history of my family split time between Massachusetts and a small island in Micronesia. So that put me a lot of time by the ocean, and the coral reefs around my island, sort of the backyard. So that got me really interested in marine biology. Through college, I did a program that was kind of in molecular biology, got me away from kind of being out and about and got into the kind of molecular side of things. And then, I guess maybe fell into or wandered into microbiology a bit. I had a chance to work for Penny Chisholm and talk to her about, graduate school opportunities. And that's, so I started my work with cyanobacteria in her lab, and that just kind of set me on this course to graduate school and beyond that, got me into microbiology and cyanobacteria. So that's sort of the short history, but yeah.
I think it's amazing. First off, where in Micronesia?
So it was in the Marshall Islands, a small island called Kwajalein. We were there for my dad's work, but it's an amazing place. If you have a chance to go, I recommend it. The coral reefs are just amazing. I'm sort of spoiled anytime I go someplace else and go snorkeling. I'm like, it just doesn't compare, but yeah.
Well, I'm a little sensitive to UV light, sadly, but I just love the idea of the South Pacific a lot. I've only been to a couple of places, and I have to be very careful when I do, but you're right. Underwater is just fabulous.
It's amazing.
So no wonder you have the interests that you do. Now, you mentioned Peggy Chisholm, and that is somebody that we're going to put a wonderful video. It's funny, the video that you sent me a link to is the same one that I was going to put up, which is really Perfect. Amazing. Yeah. And I think a lot of people, and I don't want to make this too much about Peggy Chisholm, other than point out that she was responsible for making clear the role of a particular type of cyanobacterium that most people haven't heard of and how important it is to the biosphere. Would you mind giving a quick thumbnail sketch of that?
Yeah. So Penny was really instrumental in discovering these things that we now call Prochlorococcus, and they used a special, and she talks about in detail, but they used a special instrument, a flow cytometer, to see it. And I think what's really remarkable to me was that without this kind of careful looking with this instrument, we didn't even know this organism existed. And, and that launched her career. She talks about, she just, she converted her lab to do all Prochlorococcus work. This organism is found throughout the globe. It's pretty abundant. I think we might have a slide of that showing kind of the distribution of Prochlorococcus. And even though, it can be up to like a million cells per mil or something like that. But over these vast reaches of the ocean, that's, multiply those two things together and you have a lot of cells. And so she always, highlights that it's the most abundant photosynthetic organism on our planet. And, I think it says, they're at the bottom at Prochlorococcus and its, sister genus Synechococcus. Together, they do about a quarter of the photosynthesis in the oceans. That's and, amazing. Many of viewers may not know, half of the photosynthesis on our planet happens in the ocean, half happens on the land. So it's not just forced, but really it's split evenly between those. So if you take that 25% and divide by half, you get, 12, let's say 10% of the photosynthesis on the planet happening by these tiny things in the ocean that we didn't even know existed, 30, 40 years ago. And so one of the things I like to try to impress upon my students of microbiology is that, every, 10% of the oxygen that you're breathing, right, comes from these tiny cyanobacteria in the ocean. So it's really amazing.
It's to me, it's fascinating because yes, they've been there. They've been there for 1 billion years, more. But the interesting part is that it took people to actually look carefully. And it was people like yourself and working with Penny Chisholm that were able to actually start to observe these things and think about it. What a wonderful place for you to have gotten some training, I should say. Mm-hmm. And what a wonderful opportunity for you to move forward.
Yeah, it was really, I feel really fortunate. It's kind of, I look back at my path and feels a bit of kind of bumping around and wandering. But I feel really fortunate to have that piece. I was trying to figure out where to go to graduate school, and I realized I didn't have the words for it, but I was interested in ecology. And he said, "Oh, you should go talk to Penny," and I talked to her mostly about grad school ideas about programs, where to go. And she's like, "Well, if you're interested, maybe, you could work here after you graduate." And little did I know the doors that would open and the ideas and things I would get to study. It's really, so I'm really grateful for that opportunity.
It's so amazing to me about the role of just chance in what we do. But I think it was Monod who wrote or said that chance favors the prepared mind. I'm sure it's more elegant in French, but nonetheless, it's absolutely true. If you start looking at your own life, how fortunate all of us have been here and there in just chance meetings. And that's one thing about microbiology that I want listeners and viewers to know is it's surprisingly welcoming to a lot of different people. And it always has been in my experience. And I'm really happy of all the efforts to make it more inclusive and more representative over time. And it makes me proud to be a microbiologist.
That's great, Mark. Yeah, I fully agree. Yeah.
So then you started to go through the process of becoming a scientist and get your training. And now you have your own lab. Yeah. And what do you and your coworkers, because it's really a group effort.
Yeah, for sure.
What is the area that you're kind of looking in the most?
Our lab is really kind of focused on this idea of ecology that I mentioned. So I label myself as a microbial ecologist, and I always think about ecology as kind of two sides. One is like interactions with sort of the abiotic world, and then interactions organisms have with other organisms. So our lab kind of does both. On the abiotic side, we kind of think about how things like temperature and nutrients, and then we have a big new project on iron and how cyanobacteria adapted different iron conditions. So that's one side. But the other side is looking at interactions with other organisms, that being phage as the organism. So I don't know, we could debate whether or not organism is a correct term for phage. But so we've got this great new project looking at phage interactions and trying to understand the tug and pull between those two and how they shape each other's, ecology and evolution and genomics. And I'm a little bit of a fanboy. I've got my phage t-shirt here, a T4 like phage t-shirt to represent our wonderful viruses. And that's been a fun, yes, love it. It's been a fun departure kind of from my graduate work and postdoc work to add in something cool. And I've just fell in love with viruses, just thrilled to have a chance to kind of add that into the things that we're working on.
Mm-hmm. Well, one thing that I've seen a little bit, and I don't mean to cut any of this short because I think it's good to talk about the projects you have in mind. But one thing that struck me is that the seasonality of when things are happening is really striking to me, because most people have this idea that somehow microbes are different than macrobes in terms of having seasonality. And we've seen in lots of episodes of this podcast, it's simply not true. Yeah. And I believe you have some wonderful representations of that.
Yeah. So I've got maybe a couple slides. One maybe is to highlight, it's a series of 4 graphs that are kind of highlights from my PhD student, my first PhD student, Emily, who graduated a few years ago. And this is looking at how the abundance of these different phage species, so we call them cyanophage if they infect cyanobacteria. And so we kind of drew down to look at how these different species change over time. And this is working off of a time series that I was involved with Jed Furman at USC, so just off the coast by Catalina Island. And so we just tracked how they changed over time. And we pulled about 300 kind of quote species-like things. And what she found was, they have different patterns. If you looked at the whole community, it would be seasonal. You see this kind of ebb and flow of the whole community changing. As you said, that's what's happening in the ocean. Why should it be any different, right, from things on land that we can see, the fall, the leaves changing, or, animals coming and going. So the whole community kind of changes, but as we drill down to these individual species, we saw different patterns. And so some of them have kind of a seasonal pattern, like in the upper left panel, the blue ones, and those we call seasonal phage. And then I'll jump to the one on the bottom right, which is the pink one, and we kind of wrestled with how to, what to call them, but we call them sporadic phage, is sort of the official term. But Emily came up with this great term called one hit wonders, right? Sort of like these songs that just they just show up for like 1 or 2 moments in time. And so they're kind of very spiky in their abundance. And so we kind of continued with that kind of music analogy. So back to the seasonal phage, we kind of thought of these as like songs you'd hear on the radio, like summer jams, things you only hear in the summer, or holiday music, right? Only shows up, during, Christmas time or Thanksgiving or something like that. And then the other ones, we have an upright or persistent phage. So there's some phage that just seem to be around all the time. They might bounce around a little bit, as you can see that orange wavy line. So we thought about these as, the analogy is maybe not perfect, but earworm songs are songs that are just like, they're always on the radio. It's like that Beyonce song or that Michael Jackson or, just these classic songs that you can hear almost any time of the year.
Let's just not call them earphage. I think we have to stay away from that.
That's too much. Yeah. I don't. Yeah. And then the bottom left is sort of everything else, sort of things that didn't quite fit in all those categories, things that weren't seasonal, but they weren't kind of really sporadic. They weren't really spiky, and they weren't things that are around all the time. So we just call them occasional. So I thought this was really cool that we could think about, they just have these different types of patterns. And I think the next thing to think about then is like, why, right? Why is that the case? And we think it probably connects to their interaction with their host, right? Some phage are probably very specific and can only infect a particular host. So maybe that's what's happening with the sporadic, right, phage, right? That one host or set of hosts that they can infect just show up then, and then they show up, the phage, attack it. With the seasonal ones, there's definitely, cyanobacteria, they're very seasonal in their pattern. And so, there's probably some phage that, right, are following and chasing them over time. And then the persistent ones, I think, are ones that maybe can jump from host to host, right? They can infect lots of different hosts. So as the hosts are changing through time with seasonality, maybe they have the ability to kind of attack many different hosts. So that's something we're trying to follow up with our current project, where we're kind of doing a time series analysis here, where we've got weekly samples now off of a dock with my collaborator Marcy Marston in Rhode Island, but we're also doing isolates, right? We're capturing isolates and phage and working within the lab to explore that further. Whereas this slide I'm showing is just molecular work, just looking at the sequences. So we're getting towards a massive amount of data that we're working with in that project, and we're excited to kind of connect the culture work and the molecular kind of culture independent work.
I really like that, because many times people will look at just the molecules, and there's nothing wrong with that work.
Sure. Yeah.
But you're able to work on the like, it's rather than in silico work with collected data, as the saying goes. You're also able to work with them in the laboratory and even in little cultures or plates, which is really helpful. I do want to emphasize to listeners and viewers that it's really sometimes people will say, well, why is this particular study important? And I'm a little bit nervous about that, because I have never studied anything that's terribly important to the world. But, a certain amount of our oxygen is due to these, the oxygen that we breathe every day. And you're studying the seasonality of when these particular microbes increase or decrease in number. With the interest that people have in global climate change, the idea perhaps of this issue in the Atlantic, with the Gulf Stream becoming aberrant, changing its normal, all of these things become suddenly much more important. So I just want to emphasize to listeners and viewers, this isn't just about looking at green things in flasks. It actually is, nothing is more important than the air that we breathe at this stage to us, right?
I agree. Yeah. And, we know that the world is changing, the oceans are changing. And like I said, half of the photosynthesis is happening in the oceans. Yeah, we should care about the rainforest the tundra and things like that. But the same is true for the ocean. So I think, this really kind of hits at trying to understand the system better, so we can kind of understand where it's going in the future.
I also love how, when the numbers of some of the cyanobacteria come up, then the phage are going to be reproducing in response to that. And that level of specificity is something that listeners and viewers may not get. So if you, for example, look at this model of a bacteriophage, and this is meant to be a T-even bacteriophage based on its shape, there's a particular receptor on the bacterial surface that phage is looking for. Unless I'm mistaken, it's always first order kinetics just banging into things. But when it has the right kind of receptor, it's able to stick. So there are some types of phage that are relatively broad in what they can attach to, and others that are highly specific. And so I wonder how all of that is connected to some of the changes in the phagecy. And by the way, the whole music bit with like summer tunes and winter jams, I'm really liking that.
You're right. Okay, great. Yeah. When my students said one hit wonders, like, oh, that is so great. And I was like, okay, we got to roll with it. And so, so I love that analogy because I think it helps people understand things. Mm-hmm. In terms of what we call host range. So that's kind of understanding how many different hosts a particular virus can infect. We already have some information about the different viruses out there. So the data that we're looking at is focused on these phage called myophage, although the taxonomy has now changed, so I need to update that. But these are things that are kind of T4 like the 3D printed model one and the one on my t-shirt. And those are thought to have, sort of more broad host range. So they can probably infect, a number of different strains. Then we have these other things, potoviruses, which have a short tail. They're thought to have maybe a little more narrow range in terms of the number of strains that they can infect. And then we have these cyphoviruses that have a long tail, and those are thought to be very, very specific, only a few strains. And so those that's kind of what we're trying to do with this current project of looking at isolates. And so basically what we're doing, we got maybe 50 to 100 viruses from phage from this one location, and then 50 hosts, and we're doing this giant matrix to do this cross-infection. And this is all work actually that Marcy Marston is doing in this collaborative project. So we're actually trying to ask, what is actually the host range, right? Are these rules that we've made up about myoviruses and potos and cyphos, are they the case? The weird thing is that when we do the isolations, we get a ton of myoviruses. We know that there are probably just as many potoviruses from molecular work, but we just don't get them in the isolation work, right? Which I know you're familiar with, this culture bias, right? And so we've actually been able to get a few more potoviruses and a few more cyphoviruses to kind of fill out the picture. And in general, I think it does fit this initial view that the myos are broader and the potos and cyphos are a little more narrow. But I don't think that's a hard and fast rule. I think we've got a few myoviruses, cyanophage, that are a little more narrow. But those are the types of things we're trying to understand and drill into. Back to this kind of the importance of this. Yes, there's this climate piece and understanding how the system works to kind of think about what's going to happen in the future. But there's kind of a basic science question I'm really intrigued by that, if we can call these things species, if we can try to, put some bounds on what the populations are, they're probably thousands, maybe, tens of thousands of species in this, in one kind of pocket of water at a time, or in the system as a whole. And then so imagine, thousands of hosts and thousands of phage, and then they're all infecting each other. So you have this like hairball net of interactions. And I'm just really curious, what, there's all this complexity, but, to sustain the system that still is pretty even over time, more or less. And so there's like this all this crazy interaction and infection and resistance happening. And so somewhere we're trying to do is kind of peer into that hairball and try to tease apart some of these connections. I don't know if that makes sense, but that's some of the, I think, the basic science we're trying to drill into and understand how that complex system works.
No, I think it's really relevant because I think a lot of science has been highly reductionist, where you look at one particular simple aspect of a system. I mean, that's the way I was trained back when dinosaurs ruled the earth. But in fact, the further we go into it, and with increases in the technology that we have, we can start to see how different things are interacting, because that's what life is. It's not snowshoe hares and lynxes, right? As right. Is the really fun bit that they try and hammer into ecology students. It's much more complex than that. And I understand the worry that you can get too complex. I get it. But I really admire the people that try and look into the complexity. So you can call it unraveling a hairball as many times as you want, because I'm with you.
Yeah, that's great. I mean, and that is the challenge. It is so complex. And so I understand the need to kind of narrow down and look at a particular set. And but I'm hoping that our work kind of gives us a big picture. And that connects, I think, to my journey is like when the molecular biology and the kind of drilling down into the cellular microbiology, that stuff was so cool, right? Understanding how DNA polymerase is working, and but afterwards I realized like it's not quite the thing for me. And then I didn't, like I said, I didn't have the term, but I wanted to be ecologist. I think kind of expand out and think about this, broader view of how these things all connect together, beyond just the molecular piece. So I feel, again, fortunate to be able to kind of take that molecular training and then kind of apply it to this kind of system that fits more what I'm interested in.
Several times in this podcast, we've talked about the value of bringing, and I describe it as different faces and different voices. Let's add to it different brains. Mm-hmm. The whole background of molecular biology was essentially driven by quantum physicists that learned how to be biologists.
Yeah.
Right. So I really think that sometimes the interesting questions are from people that come from outside a particular subdiscipline.
Yeah.
I remember giving a talk many years ago about some things that I did, and I had a faculty member at this other institution just insist that I was an ecologist, which would come as some surprise to the people who taught me ecology. And I kept saying, no, I'm a geneticist, and no, you're an ecologist. And it made me laugh because there's this business of putting everybody into silos. But the wonderful thing about biology, it's call it in the microbial sciences, let's call it a big petri dish, which is not entirely fair since most things don't grow on petri dishes. But again, bringing these different ideas in, and it's surprisingly relevant to everybody if you couch it the right way.
Yeah, I totally agree. Yeah, there's something amazing and sort of magical that happens, right, when you cross those disciplines, or you allow someone to your space that's different from what you're used to, or, and just a different perspective. That's so cool. Yeah, I totally agree.
Oh, no. I mean, I cannot think of a better time to be a microbial scientist than today, except maybe in 100 years.
Yeah. It does remind me of, as a sidebar is, during the pandemic, I got really interested in, well, it was sort of my way to cope is to look at the data. So looking at kind of the numbers of, case rates and things that were coming out and in my local area. And then, each town had their own kind of data report. And so I was looking at my own city and then looking at neighboring towns and seeing like, oh, these are really different. And so it made me really curious about kind of sociology and demographics. And so I end up connecting with a sociologist at my university. And we actually dug into some of this data and published a few things looking at social determinants of kind of COVID rates and looking at structural racism and, these pieces that I have some idea but didn't have, a depth of knowledge that she did. And so along with me and another biologist, we had this really cool project that we did together. It was really fascinating. It sort of scratched my itch of kind of learning about something new, but I felt like we produced something that was really interesting with our different perspectives. So as one example,
there's something in all of us, we want to impose order on life, and the whole bit from Jurassic Park, life finds a way. That's you got to do the Jeff Goldblum with you.
Of course, great, I love it.
But the fact is, if you actually look at the history of the 1918 influenza pandemic, there were all kinds of very strange things from city to city taking place as well. And trying to tease out that particular epidemiological hairball. And by the way, I'll be using that term hairball a lot because I like it. Because it's messy and disordered looking, but there is structure to it. So hats off for trying to find a way to do something during the midst of that bad time that we all faced.
Yeah. It was really, it was a fun kind of side project and also, just enriching to learn something new. So,
can I take a moment to ask you a question about cyanobacteria? And I'm just throwing it this way. I noticed you had some slides showing the different colors that they have.
Yeah.
And I would love it if you could just take a moment to remind people what those different colors mean, because I find that fascinating.
Yeah. So, like you said at the beginning, these were originally called, the larger phylum, which is a giant group of organisms, were originally called blue-green. And most of them, I think, are tend to be green. There's kind of a bluish hint, I would say. My wife is a graphic designer and color theory nerd, so I get her opinion on it. But I think it's a little bit of that bluish tinge. But for the slide that we're showing with all this kind of spectrum of colors, these are just the Synechococcus and Prochlorococcus organisms that we've been talking about. And actually, the picture on the left with the really kind of thick, colored bottles, those are all Synechococcus. And so they have this range of different colors that they can have. So they all have chlorophyll, right? Which is, all nearly all photosynthetic organisms have. That's the same molecule found in leaves that does photosynthesis there. But on top of that, they have other pigments that they use to capture light. Because chlorophyll only captures light at, particular wavelength, like 660, 680, something like that. And that's the wavelength that it works. But then if you can add on top of that these different pigments, then you can capture light at other wavelengths. So that's really kind of, in essence, what's happening here. So these cyanobacteria have these other things called phycobilisomes. There are these protein structures, and they look kind of like an antenna, like just kind of fingers sticking out, and they bind other pigments to the protein structure and basically funnel light down into the photosystem to where the chlorophyll is. And it just gives them different colors. It's, in some ways similar to leaves here. So in North America, the fall is coming and leaves are changing. So we're losing that green color. And I think hopefully everyone's taught you in elementary school that, the colors that show up are these other pigments that these plants have. That's always there, but you need to get rid of the chlorophyll to see those other colors. And so, in essence, that's kind of what, cyanobacteria are kind of doing the same thing that the leaves are doing. They have these other pigments to absorb light at other wavelengths.
I was going to say, and this is not something I believe you work on, but I have to ask you since I've got you here. Have you seen the paper where there's a claim made that there's a cyanobacterium that's able to harvest near infrared light?
Yes. These are the cave cyanobacteria.
And also at hydrothermal vents with the idea when the hot water enters the cold, it generates a small amount of light.
Yeah, I haven't read that paper in detail, but I scanned through it and I think it's so amazing. I'm like, I want it to be so true. I don't know if there's been follow-up studies, but it makes sense. It's just like there's this niche of light at this spectrum, right? I mean, I think the cave cyanobacteria paper, I don't know if it's cyanobacteria. Yeah, I think it is. Of they're absorbing light that can bounce through and then has this particular wavelength that's filtered through the cave, like. Why not at the bottom of the ocean where you have this other wavelength? I mean, it reminds me of pit vipers, because pit vipers, right, they can see infrared, right? So they have these organs that sense infrared, this light that we cannot see, right? And they're going through this world, seeing the world in a way that we don't. Now that's kind of sensory, but same idea with energy. Like, why not? Why not? If you can figure out the proteins to do it, why not be absorbing this light this wavelengths coming off of kind of heat from the vents.
We had Hazel Barton on the podcast about cave microbiology and talked about this topic. I just find it fascinating. And it reminds me of the fact, and I want listeners and viewers to remember this, that microbes are very good at living at what you and I would call the margins of everything. Mm-hmm. To the point where you can easily find microbes that can live off the impurities in agar. For example, no added carbon source. Yeah. I have had that happen to me, which is no fun. When you're not expecting it as it is. But what I'm getting at is the same idea of taking advantage of every single photon of light that you can. Yeah. And it's interesting because I had an introductory student ask me this question: if the colors that we see are reflected light, why are there no photosynthetic organisms that are dead black? That is to say, absorbing everything. And I really couldn't it's more of a philosophical question, but I really like it. Maybe that's an extraterrestrial organism.
Yeah, right. You gotta catch it all. I'll dare to venture a guess. My thought is, there's in biology, we always talk about trade-offs, right? You can't, some microbes are can be good at doing lots of different things, like Rhodopseudomonas palustris, right, is organism I work with a little bit for one of my postdocs, and it can do lots of different metabolisms. But in general, you have to pick and choose, right? So maybe you can't make enough pigments to cover the full spectrum. That would make you black. And
I was just going to say that's what I loved about the illustration that you had with the colors, because that's what I wanted you to be able to tell listeners and
Yeah.
viewers a little bit about.
Yeah, sure. We can go back to that briefly. So, the different colors I think are going to reflect the different environments that they're in. So the green one, all the way on the right, those you tend to find these green strains in coastal environments. And so if you've been to the ocean, been close to the ocean, you can see like the water often is kind of greenish, maybe brownish because of, the things that are flowing in from the coast, and it's highly productive. And so because of that, certain wavelengths get kind of absorbed away. And actually some of the algae that's there takes some of that light away. So what's left over, right, is, the light that you can use. And so the green colors reflect that. As you tend to move away from the coast, the water gets bluer. And if you ever had a chance to take a boat as far away from the coast as you can, and you get out, the color is just this brilliant azure kind of blue, blue. Some people call it oligotrophic blue. And so the light quality changes, right, in the environment. And so because of that, the organisms, the cyanobacteria, adapted to different light conditions there. And so as you tend to do that, you go offshore, you tend to shift towards kind of those reddish, pinkish colors on the left. And, there's variation in between. So you have, a little bit of like some are a little more orange, some are a little more pink. And some actually can change their color a little bit, kind of like from a pinkish to an orange color as well as an adaptive trait. So this is called chromatic adaptation. And then the other extreme is with Prochlorococcus, which on the slide are the ones so the picture on the right, they're kind of in the middle. There's a few there that are kind of this greenish color. And those are kind of break the model of what we talked about. They're not like the pink Cyanobacteria, but when you go to the open ocean, right, they are really adapted to pick up blue light, which is kind of predominant, especially when you get deeper down in the water. So as you go down as well, the light quality changes. So if anyone's been diving, that the colors just, with your own eyes just change. They get kind of muddy, for example. So the wavelength's going to change. So Prochlorococcus, they don't have phycobilisomes. They don't have those extra pigments, but they do have chlorophyll B, divinyl chlorophyll B, which allows them to kind of pick up light in the blue kind of range. And you mentioned, microbes kind of eking out at the edge, picking up the last few photons. There are some Prochlorococcus that live really deep, like 200 meters down, and they have a lot of chlorophyll B, a divinyl chlorophyll B, and so they can pick up those, those blue photons right at the bottom and just kind of eke along. They're just kind of chunking along, doing what they can. So that's also another cool area, is people discovering really interesting Prochlorococcus that live down there, and also in anoxic zones, and they're just they're eking away. They're living there, sucking down all the light that they can.
Right. And I mean, we don't have time to talk about that one event where we link photosystem 1 and 2 that makes all this possible. Yeah. We won't do that today, but because I really want to hear more about some of the projects, especially about iron, because I have lots to ask you about that.
Yeah,
did I hear you correctly that you received an NSF career grant for this?
I did. Yes. That came in the summer. So thank you. Yeah, I'm really, again, I feel really fortunate to have that opportunity. And yeah, so we're going to do some great science the next kind of 5 years, and then also some great education and outreach kind of work connected with that. So yeah, so that's focused on the iron kind of side of things we're looking at in the lab.
I like talking about the iron aspect of it because I'm always with my students, I'll ask them things like, what does a bacterium use iron for? And they just kind of look at me and say, well, for their hemoglobin. And I'm not making fun when I say it. I mean, you've heard me talk about eukaryocentrism and oxycentrism and colicentrism. And these are good examples of it. And I want there to be a microbiocentric view of things.
Yeah.
And so the iron is necessary for things like cytochromes. Would you agree with that?
Yeah, there's a heavy use of iron in photosynthesis. So kind of shuttling electrons around the photosystem, making the components of chlorophyll, things like that. So so that's a lot where it's happening. But every cell needs, I think everything on life needs iron. It's a really important ion that sits, as a reaction center for a lot of different enzymes. But in particular for photosynthetic organisms, there's I think a heavier burden for it.
And with the change that took place with oxygen entering our atmosphere in large amounts, it affected the availability of iron, which is what you would like to talk about, surely.
Yeah, for sure. And it maybe as a quick aside, like the evolution of different enzymes. I'm not an expert at this, but as the oxygen changed, organisms had to, they kind of evolved or merged in different metal conditions. And so cyanobacteria are kind of like, they're a little bit stuck in sort of in the ancient world of where there's plenty of iron around, or available iron when you had less oxygen. But in things like diatoms, other phytoplankton that do photosynthesis in the ocean, they showed up way later, right? And so they have evolved kind of enzymes that use other ions, like I think zinc and cadmium. And so there's this cool kind of picture of how evolution has changed because of what you kind of grew up in, right? The milieu of metals that you grew up in. So that's an oversimplification. I'm not expert, but it's a cool picture. So I think this is hopefully true. People can comment and correct me, but cyanobacteria kind of are in some ways kind of stuck, or they evolved with kind of using iron for a lot of these enzymes. And so because it becomes a really important nutrient in the ocean, not just for cyanobacteria, but for all phytoplankton. So some other numbers around, about 30% of the oceans are limited by iron. So that means they don't have enough iron to grow. They've got all the other nutrients they need. So if you added iron, the cells would go bonkers, right? They just grow like crazy, or they grow more. And so iron turns out, this picture of thinking about photosynthesis and global carbon and kind of these patterns, iron, it becomes an important player in that. And so the overall project that we're doing with Synechococcus is kind of the cyanobacteria is trying to understand a better picture of how they're adapted to iron. So there's been a lot of focus on these low iron places that often have low iron amounts, and it's chronically low iron. But that's not the whole picture. Every cyanobacteria has to use iron. They still have to find it in those other places. Maybe it's higher iron, right? But maybe there's mid-level iron, right? Or maybe there's places with low iron, but it's very, very sporadic, right? It's very variable, right? Sometimes there's a ton, sometimes a little. So we're trying to paint this kind of landscape that it's not just this one kind of iron condition, but this, larger, these, different niches that they have to occupy. And what's really nice about Synechococcus, if we went back to that map of where they're found, they're found everywhere you go in the ocean. Really, you could find some Synechococcus or Synechococcus-like thing from the equators to the poles, coast to the open ocean. So we have this great system of organisms where we can, look at how they're adapted to iron in all these different parts of the world. So that's kind of the major pitch of kind of understanding that. And I think the other thing that I like about the project is connected to what we were talking about before, is we're going to work with cultures in the lab that we've isolated, but we're also going to do some field work and do some molecular work, both in the field and with cultures. So kind of trying to bridge just culture work and molecular work at the same time, and to hopefully give us a bigger picture together with both of those tools.
When I was in graduate school, it was interesting. There was an ecologist, he's still with us, Paul Ehrlich, who was a character. And there was another professor who did microbiology, Charles Yanofsky. And so what Paul Ehrlich would do during his seminars is show a slide, and it would show Charlie in front of a fermenter. And he would say, here's a microbiologist in his normal laboratory. And Paul Ehrlich worked on butterflies. And then he would show the mountains in Colorado, going, and here is my laboratory. Yeah. And this always offended me because I don't mean offended in that I was all taken aback. It's not entirely fair. Look at your own work. You're able to do plenty of field work as well as the bench work. And this is more and more what we see. And it's important for listeners and viewers to know it's not like you might see on television with the glasses and the white coats, nor is it like Indiana Jones.
Yes. Yes, unless you're that I'm forgetting their name, but the cave microbiologist I think that you talked about.
Hazel. Absolutely. Yes. Hazel. Wow, is a force of nature. That's so amazing. There you go.
Yeah, but yes, we get to do both, and and that's part why I chose this field to have the opportunity to go out on ships and be on the water, and
Yeah, we've had a number of people that have, because I've always been interested in marine microbiology, and I think it's very interesting because I'm used to just looking at salty plates, and then you folks go out on. I've never been on a cruise, which is good, because probably I wouldn't mix well with it. But it's a wonderful opportunity for your coworkers and students. And you have all different kinds. You have undergraduates, and you have graduate students, and you have postdocs. Am I correct?
Yeah. We got the whole range. Yeah. Full meal deal.
And you have outreach to a primarily undergraduate institution as well. Yeah,
we're truly, I mean, the undergraduate piece, I know you appreciate, teaching undergraduates is really rewarding and important. And it's a big focus for our university at Clark. But we also have the graduate program. So really kind of span this range of things. I love it. I think back to thinking about like bringing different voices and different experience. And I think the piece that I love working with undergraduates is they're kind of wide open, right? Their minds are, why not have a microbe that is right, has all the pigments, right? Can it absorb all the wavelengths? Mm-hmm. So I love that kind of open-mindedness that brings us energy and curiosity.
If you've ever, and I am revealing something about myself, I went through a period of time when I was an undergraduate where I was studying Zen Buddhism. And D.T. Suzuki has this expression, there's a whole ideogram for it called the beginner's mind. And that sounds like an insult, and it isn't. A beginner's mind is open to everything.
Yeah.
An expert has already decided lots of questions aren't worth considering.
Yeah, that's right.
And I cannot tell you the number of times that a student who's new to microbiology has seen something super unusual that I didn't see, because they didn't know what to look for or not to look for. So that's why I'm really glad that you have some of that in the mill that is your research group.
It's great. I love it. It's a really fun part. I don't know if we do have a slide, I think, up of, as an example, I've got some photos of undergraduates working lab. One is a photo of us working off the dock to collect some of these cyanobacteria. And as I was pulling this together, I realized, and it's paired with some photos of green and red strains that we get in Narragansett Bay in Rhode Island. And Cammy's there. And I was looking back, we did not plan that. We were wearing red and green shirts at the time to reflect the two different types of cyanobacteria that we didn't realize, but we would find in there. And then I also am wearing a red mask as well. So as my wife was pointing out, you've color coordinated this photo connected to your research. That was serendipitous.
I think it's great. You now have team red and team green. I mean, this is the way to do things.
Yeah, it's so great. Yeah. And like you said, that's the fun thing part is that the colors, right, are just so amazing. I think it adds another kind of dimension to this that really makes it fun.
So we talked a little bit about the type of light that cyanobacteria can use to drive photosynthesis, sometimes very, very, very low wavelengths of light, almost infrared. Mm-hmm. How high can they go?
In terms of intensity?
In terms of could they actually get a benefit from ultraviolet light?
Ultraviolet. That I don't know offhand. I think not. I think their pigments are tuned, to more visible light. But I think that's another space where, maybe there is some microbe that does capture UV light. I think the challenge is UV light is damaging, right, for DNA in particular, but other things. So that higher energy, it's again that trade-off of like, there's a cost, right, to be able in an environment where you could use that. You probably want to be making molecules to block that or defend yourself from that. That's a great question.
I have to laugh a little bit because I wonder. I'm certain people have looked, but we never see the negative results. You know what I mean?
Yeah.
And then remember that Deinococcus radiodurans was first isolated from food that had been sterilized by gamma radiation.
Yeah. Yeah.
So I think it's something to think about anyway.
Yeah.
Life finds a way.
That's right. Great. Bring Jeff Goldblum. I try to, if I could bring Jeff Goldblum in every lecture, I would. There are cyanobacteria that live in these like dry mats in Antarctica and other arid places that are probably getting hammered with UV. And so they probably have adaptations to that kind of environment. So
interesting. One thing we haven't talked about, and it's part of your website, is you do a certain degree of 3D printing. And you know that when I say I do, you understand I mean Dr. Jennifer Quinn does it, just so we're clear.
Yeah.
And I notice you have some models of things that you have actually made, and you have links on there to where there's a repository, I believe the NIH runs it, of many different types of things.
Yeah, that's right. Yeah, so I kind of got into this as a little bit of a hobby, but as a kind of a teaching tool to try to visualize things. So I'm always trying to think about how to explain things to students in a better way, and learning in a visual way is really important. So I print these things and bring them in the classroom. During the pandemic, I printed a giant spike protein and antibody that binds to it to really kind of demonstrate these molecular interactions. So some of it's fun, some of it's teaching. But those, the NIH does have a 3D print repository. So if you build a model, you can put it there. So I've tried to put a lot of these molecules up there. But these molecules that I build, I don't do the hard work of it in terms of figuring out the structure. So people doing X-ray crystallography, they figure out where all the atoms are located for a particular protein. And you can download those through the PDB, the protein database, and then basically put them in a format you can 3D print. So that's kind of the little picture of how that's done. So that's so one of the things I'm doing on my sabbaticals is playing around with some of those a little bit more, and as a bit of a hobby. But my plan is to print a giant cyanophage to have when I bring back. And since I'm here, I'm kind of remiss that I don't have any of my 3D print toys because I left them back. So I'm in my mother-in-law's house because it was a quiet space to record for today. But you can see some of those online. I don't know if one way maybe to connect to some of those models and to the iron thing is that I do have a model for a ferritin protein. So this is one of the ways that cyanobacteria and other organisms can deal with varying iron, and is that when it's basically a protein shell that stores a bunch of iron atoms, and so the proteins fit together, and there's a little space inside, and so it's like a little cache of iron that they can store. So if the iron conditions get low, then they can kind of pull the iron out of there, like a little stash of iron. So I found a protein model for one that's found in Synechococcus and printed it out, and it's just I think it's so fascinating at that molecular scale to see how it works, right? How they interact and how you can make these kind of they're basic simple shapes, right? And they do these cool things for the cell at this molecular scale.
So my wife's a mathematician, and so she says it all comes down to mathematics. And I don't know if I have a 3D print behind me of a Mimi virus, for example.
Yeah.
And that took us some time to find. But again, I'm going to put a link, of course, to your page, but also to some other sources for folks interested in 3D printing various biological molecules because or structures. It's very interesting stuff. So thank you.
Yeah, that's great. And so I do have that animation of the ferritin build that maybe we could look at while you have that up. And the mathematics is another piece that's really interesting to me. So the phage capsids, I'm sure you and your wife appreciate like the geometry and how you can build those. And you mentioned the giant virus, the Mimi virus. And that's a fun thing I bring into my class as an extra credit assignment. I think similar to some of the things you do of like getting students to make their own art. So I have them look at T number for icosahedral phage capsids, and do some math and kind of figure out the rules a little bit. And then they have to build a artistic representation of a phage. That's been fun. I get all these cool things. Again, they're up on my shelf, so I can't show you them in my office back in Worcester. But I had someone make a gingerbread capsid, and put the pieces together with icing. Some made some out of clay. Toothpicks and gummy candy, and so it's a fun way to get the students to kind of think and explore the phage biology in a different way.
It's kinesthetic too. There's something about building it, and anyone who is interested in going to dental school, part of that process is learning how to actually build things, and I think it does help with understanding. Yeah,
it does. And one other thing that fascinated me is I had students kind of choose it was in my virology class, I had them kind of choose some molecules that they wanted, some virus that they wanted to build. And so we printed some, and then we put them together. And then I realized the way they fit is just so amazing that I don't think you can really appreciate on a computer screen. And then just having them, that tactile piece, and seeing they just kind of lay on top of each other in this pattern. And then, we put them together, and I was pushing on it, and I was like, you could probably stand on this 3D printed thing, and and the way the capsid molecules fit together, it would not break. Now, obviously, it's not the same as it is maybe on the molecular scale because they're not exactly. Those molecules aren't really rigid like a 3D print. Dare I say they're probably squishy in terms of their structure, because the atoms are all moving around a little bit. So so I think your producer or podcast person, Dave, would say this thing, like they're kind of like squishy Lego, which I think is amazing, right way to think about it. Yeah, they're, in reality, if you could zoom in, that's probably what they're like.
That's also a good band name. So, Squishy Lego, that's for you.
All right, I'll take that, yeah.
I have another question for you, though, Nathan. We were talking a little bit about, and I love this expression, a symphony of bacteriophages that are confronting these cyanobacteria during like rises and falls that they take place. Yeah. Is that connected with their ability? Well, on one level, it has to be, but is it connected with their ability to make oxygen?
Yeah, I mean, it should in some level, because really, I mean, in a typical lytic phage scenario, they end up killing the bacterium. So if that organism dies, it doesn't make the oxygen. On a larger scale, though, I think there's a bit of a balance that's happening. So we know in the open ocean where the bacterial levels are kind of constant throughout time if you go in a tropical region. So what kind of happens is all the bacteria, or the cyanobacteria, and I guess most of the bacteria, they double once a day, about, on average. And then that also means that their total abundance stays the same. So they all kind of die once a day. So there's this, every day they're doubling and then getting killed. And so, who's killing them? It's protists eating them, but also phage. And so there's like this interesting balance, like they're constantly growing and dying. So even though, right, we think about phage as killing these organisms, and that's maybe a negative thing, it's just kind of how the ecosystem works in that scenario where it's kind of even. I think that's really fascinating. And this is where I think about this, like the ocean is constantly churning, is the way I think about it, is they're all kind of constant through time on this, small scale, but there's all this stuff going on. And that's, again, where we're trying to peer into this kind of hairball to think about who's infecting whom, what's happening underneath that's something that's relatively stable. If you go to other places, though, where you have a strong seasonal cycle, where the cyanobacteria come up in the spring, which they do in Narragansett Bay, the phage are going to be chasing them, and they're probably going to, control their total abundance and control the oxygen. So there is a bit of play there, but I think of it as more of kind of a balance, right? Rather than just like this boom bust.
We are swimming in a microbial sea.
Yes. I would be, if I can add one more thing, I think I would be remiss if I don't mention that this fascinating thing about cyanophage regarding photosynthesis and oxygen is that cyanophage have done this interesting thing where they've captured a gene from their host. It's photosystem genes. And so when they are infecting the cyanobacteria, they actually turn on this phage version of the photosynthesis gene to keep the cyanobacterium doing photosynthesis, to basically make energy to make the cyanophage, right, continue the infection and production. So it's just the insidious, clever solution that they've had. And so in some ways, it probably is helping continue oxygen production, or at least producing energy. And so this gets in this cool aspect of the coevolution of these two organisms. And they've picked up other genes from the host to pull in phosphorus into the cell, or to deal with nitrogen. And so there's this really amazing evolutionary story of these, of phage kind of co-opting these genes. And one other thing, I think maybe to fit with this theme of the symphony, is that I talked about the cells divide and then they get killed every day. And the cyanobacteria, right, they're synchronized to the light cycle. So they grow during the day when there's light and energy, and then they divide sort of in the middle of the night, early morning. And it shouldn't be surprising, but the phage have a synchronicity as well. So they are, they're infecting at certain times of the day when the cells have energy. So they're both tuned together. So again, I think this idea of symphony, they're working together, and it's just this beautiful kind of piece of music and interactions that are happening in the ocean.
So it's not rise and shine, it's rise and lice.
Yeah. Yes, that's great. Oh, I love it.
So where do you see your work going the next 5 years, since you've now got some new things? What would be your perfect view?
Yeah, we're just launching this iron project. I mean, we've done some iron work. That's been kind of bouncing along, but now we really have the funding and support to really kind of expand that. So I'm excited to kind of build that out. And the sort of it's my hope with that is to build this grand opus. So we're going to collect all this data, these different parts of the world and different isolates, and then hopefully maybe write this cool paper that kind of looks at, the landscape as a whole and pull out kind of like some common themes that we see across these different parts of the ocean.
I was just going to say, we often make a joke about forest and trees. And it's really common for most of us to focus just on the trees, but the overall shape of the forest is important too.
Mm-hmm.
And so it would seem to me like the project that you're describing is a good way to look at patterns on a larger, if I may dare to say, planetary level.
Yeah, exactly.
As opposed to what's simply in, say, a 50 ml flask or a petri dish.
Yeah, that's exactly the goal. I think just try to broaden our view, capture all kind of the important parts of the ocean. And so it's ambitious. Hopefully we'll get there, and hopefully we'll get or 80% of the way. With the virus work, I mean, I think we're just collecting. We've just got a ton of data now about looking at host range, and then we've got this time series that now we're getting the data back for. And so I think there's going to be really cool things that we're going to do to connect those two, to say, we've got this particular phage that only affects, a few things or broad things. Then we can look into the time series data and say, what's its pattern? Is it a one-hit wonder? Is it a seasonal phage, right? And our prediction, right, is if it's a broad host range, it should be a persistent type. It should be there around all the time. So we're going to get to peer into that, as we've talked about that hairball, look at those parts of that hairball, the connections and bridge between those two. Some of the things we're finding, and I wish we had more time, but we're finding interesting things where sometimes the Cyanobacteria, Synechococcus that we isolated, there's a phage that came along inside there, and they appear to coexist for stably, which maybe isn't too surprising. But at first we're like, oh man, we've got these cultures that are contaminated with phage, a good number of them. But that has been, I think, a serendipitous kind of discovery. And so we're exploring that coexistence. That will be the next grant and the future in maybe the next 5 years of understanding that. And it's not a new idea, but what we're finding is that when we have a host and phage together that we pulled out from nature, if we try to throw in other phage, it doesn't get infected by those, right? So there's this sort of like lysogeny, right? It's sort of that they're protected against it. So there's really cool things to explore there. We know that some closely related strains that we have of Synechococcus, some are really resistant, some are really susceptible, and we know we're starting to see that has to do with probably proteins that have to do with the surface of the cell. So that attachment piece that you're talking about with the phage finding the surface, but some are probably host defense genes. So there's, I think, a lot to explore there in terms of the coexistence, but also the host range. What are the genes controlling that? So there's a lot to play with in the future for that phage host system.
I give a lecture to my students. Remember, I only have one microbiology class here. There's just the one.
Oh, wow.
And I call it the heresies of microbiology. And these are things that most people don't think of when we talk about microbiology. And one of those things is that most microbes don't live just with themselves or their progeny.
Yeah.
They live in interactions with other organisms.
Yeah.
And that makes them hard to study. That may be what microbial dark matter is really about. But it's interesting to me to remind people it's not about single colonies and pure cultures.
Yeah.
That is the way you can answer a particular set of questions. And I'm glad that we did it that way. And I certainly did it that way. But that's not how they normally live.
Yeah.
As I often say, the microbial agenda is not ours. And we really think it is.
Great. Yeah, that's great.
And so this is what I like about the work that ecologists like yourself are doing, because you're looking at groups of organisms actually.
Yeah. Yeah.
And it's really relevant to the world, especially with the ideas of climate change, especially with the idea of further climate change independent of human actions as well. Yeah. There's all kinds of things to think about. And what you're starting to do, if you don't mind my saying so, is come up with some baselines to understand these things. Keep in mind that when I read about in textbooks about phage interactions with bacteria in the ocean, it's mostly described as whenever there's a whole bunch of one type of host, there's a phage that knocks it back down again.
Yeah. Yeah.
I appreciate that, but that's not really what's going on. It's much more complicated. I believe you discussed the Red Queen yourself in one of your papers.
Yeah. So this is another kind of piece that's come out of working with Jed Furman and the time series he has there, the San Pedro Ocean Time Series. And so in this case, this is all kind of the metagenomic world, kind of looking at the sequences. And this is work Cesar Ignacio Espinosa. He's the lead author. He really kind of did this hard work and wrote out the story. But and he's now new faculty at the Keck Graduate School in Southern California. Cool. So this is a case where this came out before we kind of came up with these one-hit wonder and persistent phage. And so when he looked at the data set, he kind of was interested in these persistent phage, like so these phage that are found kind of all, throughout most of the samples. You can see them. And then what he did was kind of look at those, dig into one of those species or persistent things and look at the small changes, so individual nucleotide changes over time. And what he found was that it does have the kind of like you're describing of like one strain comes up and then it goes down and then the next one comes up. So thinking that there's a particular, strain of phage that attacks one particular type of host or a set of hosts. And then once those go down, then it makes space for the next phage to come up. And it's like this constant waves of strains within this species that are showing up. And this is, fits exactly what we kind of expect for this Red Queen hypothesis that's been around for a while. It comes from, Alice in Wonderland, and how, the queen runs faster. I guess it's Alice is running and then the queen is chasing her or vice versa. So you're constantly moving to kind of move faster to catch up. And so it's this idea that, potentially the ecosystem is evolving. New strains are kind of showing up to kind of chase the thing that evaded you, a few weeks ago. I don't know if it's evolution happening or if it's just their existing strains that are chasing each other. I'm not quite sure how you tease that apart in a complex system, but it was cool to see this result. It fits this kind of theory that we have. And I think that's one of the other goals we're trying to do is think about getting some hard data for these ecosystems to describe what's happening so we can see, does it match with the models? Does it fit with the models? Is it this Red Queen hypothesis or is it some other, piggyback the winner, right? Or kill the winner or, these other models. So, yeah.
Now, as we start to kind of go toward the end of the podcast, I happen to know that as we speak, you're about 35 miles south of me.
Yes, that's right. Yeah.
And you're doing what's called a sabbatical.
Yeah.
And that's what happens I don't know if it's for you every 7 years, something like that usually, where you have some time off to recharge your batteries, learn new things, do new things. Would you care to share what you're up to in Olympia?
Yeah, sure. The main thing for the sabbatical for me kind of professionally is to, honestly, to catch up on writing some papers that I've been sitting around. I think, teaching, I teach a fair amount more so than other universities where we're kind of, Clark University is sort of, we call ourselves an armadillo or a chimera or unicorn, pick whatever your example. But, sitting this kind of like, we're not an R1, we're not quite a small liberal arts college, right? We're in the middle kind of doing, both, trying to be both excellent at kind of that spectrum. But so that just means I teach a lot more than other, universities and, which I love. But also means I sometimes just have less time to write. And so I've got some projects from students, undergraduates and master's students primarily, that I'm trying to catch up and get those out. And as well as start to think about writing these new papers, especially with the phage data that we're getting in. So that's the kind of professional piece, but, on a personal level, just trying to take some time to, unwind a little bit, and try to think some great thoughts and spend some more time with my family enjoy the great Pacific Northwest, do some foraging and shellfishing and fishing. And so that's kind of what that looks like. But really for the professional side, it's really kind of writing, a lot of kind of writing and yeah.
Well, you're going to have to come up and visit. And
I will. I would love to.
And we'll show you some weird stuff in the ocean up here. Yes,
I'd love to. Yeah, I've been following your podcast and hearing about these, different microbes you're trying to pull out from the Tacoma area, and I also just want to look at all the little trinkets and things that you have in your office. I could spend an hour probably just doing that. That would be really fun. And then obviously to talk with you about microbiology and teaching, and so yeah.
If you go to the trouble of driving up here, I'll take you out to dinner. It'll be nice.
Okay, that sounds great. Yeah. I love Tacoma. Actually, I've been a few times, and it's there's a great art museum. It's in some ways it reminds me of my my city where I'm at home in Worcester. It's kind of mid-sized city, and so I look forward to that.
By the way, I'm really glad you pronounced the name of the city for me, so that I'm not going to mess it up too much.
Yes,
Worcester.
Worcester.
Right?
That's right. Worcester.
That's it. Well, Nathan, thank you so much for this fun episode. And I hope that everyone will take a moment at the conclusion of this and take a deep breath and realize that they owe it to cyanobacteria. I wish you and your family the very best, and I hope to talk to you soon.
Thanks, Mark. This was a lot of fun.
Appreciate it. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with awe-inspiring links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Nathan Ahlgren is in the biology department of Clark University in Worcester, Massachusetts. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.